So, we had a brief introduction to the autonomic nervous system. So let's go on now to look at each in turn. And we'll start with the parasympathetic nervous system. I'm going to put a few clinical cases in as we go through the lecture course. Who can tell me what abnormalities this cat is showing? Sorry? Dilated pupil? Which side? It's left. Good, well done. Yeah, always refer to the animal by its things and not the way it looks to you. Okay, the pupil on the left is bigger than the pupil on the right. Is that definitely a dilated left pupil? What could it be? Could be a constricted right pupil. Any other abnormalities around the eyes that could give you a hint as to which of those two is the case? Yep, so it's prolapsed effectively. It's prolapsed across the eye. So, prolapsed third eyelid, nictitating membrane, and a pupillary irregularity. Any more abnormalities? The right eye is more closed. Yeah. Typically with this syndrome, you get a drooping upper eyelid. And if you look very closely at the profile of the left upper eyelid and the profile of the right upper eyelid, you can see that the right eyelid in the sort of central area there, just above the pupil, is just kind of drooping a little bit. Yeah? So, drooping upper eyelid, prolapsed nictitating membrane. Are we happy that it's the right eye that's abnormal? Yep, okay, constricted right pupil. And the fourth clinical sign you get with this syndrome is typically an exophthalmos, i.e. the eye is bulging to some extent, but you don't really see that very easily in this case. We talked about the functions of the autonomic nervous system last time. Basically, we are talking about homeostasis. We're talking about a system that looks after the internal workings of your body so that you don't have to worry about it too much. We talked about the characteristics of the neurons of the parasympathetic system. I'm not going to go through it again in detail, but the fact that I've put the slide up twice should tell you something about its importance. This is a diagram of the parasympathetic outflow. You'll remember in the last lecture I was talking about it being a craniosacral outflow. And this is what I mean. The bulk of it comes from the cranial nerves, and then some of it comes from the pelvic nerves. It innervates the viscera throughout the body, and the way it does that is by cheating, in inverted commas, in that the outflow, particularly from the cranial outflow, but also the pelvic outflow, the sacral outflow, doesn't restrict itself to the dermatomes or the body segments that it comes from. And so it crosses many dermatomes. And the outflow from the 10th cranial nerve, the vagus nerve, innervates parasympathetic outflow all the way down as far as the small intestine. So your vagus nerve is coming all the way down through the neck, down through the thorax, down into the abdomen, and innervating the viscera as far down as the small intestine. And you'll see that clinically. For example, cattle get a condition known as vagal indigestion, where they have an abdominal lesion that is affecting the vagus nerve and is altering their gastrointestinal function, altering their ability to absorb nutrients. With the exception of 3 ganglia, cranially, the postganglionic nerves are small and in the tissues. And if you think about it, the 3 ganglia are really quite sensible in the sense that they are ganglia of the head. The ciliary ganglion, the pterygopalatine ganglion, and the otic ganglion. And if you think about it, in places in the head where the anatomy is quite constricted, and especially in the eye, the last thing you want is an extra little set of nerves getting in the way in the tissues. You know, if you think about the eye transmitting light, the fewer cells you've got in the eye, the less it's going to get in the way of the light, the better vision's going to be. So it makes sense in a structure like that to have a little ganglion out to the side where all the extra neuronal cell bodies can be. But throughout most of the body, where size is not really a limiting factor, then it makes sense to have these small networks of postganglionic nerves that are actually in the tissues that are innervated. Parasympathetic function in somatic tissues. Parasympathetic there in very heavy inverted commas, because this function that I'm talking about now is not part of the parasympathetic system. Blood vessel tone is under local control, and nitric oxide plays the role of the parasympathetic mediator in that network. It's responsible for controlling the tone of those blood vessels in response to an awful lot of things going on at local level. Whereas overall perfusion is controlled centrally via blood pressure, and that is under parasympathetic control. So the parasympathetic system sets the level of perfusion for the whole body, and the local control centres control the proportion of perfusion taken by that individual vascular bed. If something exciting happens, the sympathetic system is able to override that in a crude way and divert perfusion to the areas where it's needed for flight and fight. I talked about the outflow of the autonomic nervous system being intermediolateral, and you can see this if you look at the cranial nuclei in the medulla. This is a section of the middle of the medulla oblongata. The structure in the middle here is the fourth ventricle, and the cranial nuclei are around it. You can see from this that we have got the sensory nuclei, like the sensory nucleus of the fifth cranial nerve and the nucleus of the solitary tract, dorsally. The motor nuclei for the cranial nerves that have motor function are ventral, and there are two of them because in the cranial outflow there are two different sorts of somatic musculature. And the autonomic nuclei are in the middle, intermediate between the two. And that really is the take-home message from this diagram. The 3rd, 7th, 9th and 10th cranial nerves have parasympathetic outflow, and it comes from an intermediate part of the cranial nuclei. Here again are the neurotransmitters in the autonomic nervous system. For example, if I asked you a really dull and boring question like, describe the alpha-2 receptor, you could tell me where it was, what it did, and give me an example of a veterinary drug that was an agonist or an antagonist at that receptor and what it was used for. I want to talk a little bit now about Otto Lowy, who was a very famous scientist at the turn of the last century and won a Nobel Prize for his work in 1936. He's the man who discovered the chemical nature of the synapse. And the reason for talking about it now is because he did it by studying the parasympathetic nervous system. In Lowy's time, it was known, and had been known for almost 100 years, that nerves worked through the transmission of electrical impulses. It wasn't really understood how they did that, but it was known that electricity in some form travelled along nerves. And it was also known that this electricity crossed synapses and that there was some kind of delay at synapses. There were two schools of thought. There was a school of thought that said that synapses were some kind of electrical thing that transmitted the electricity across from one cell to another. And there was a second school of thought that said that they were chemical, and that somehow the synapses converted the electrical energy to a chemical energy, transmitted it across to the next cell, and then converted the chemical energy back to an electrical energy. And that was thought to be just a little bit avant-garde by many workers who were sort of firmly in the, why would you do that? Why not just have an electrical connection? But it was beginning to be believed, and there were various reasons why it made sense, not least the delay in processing that you get at synapses. How do you do that delay electrically unless you've got a capacitor the size of a house to store the electric charge? And look, they're only small. There must be something else going on. So Otto Lowy had a dream. And the night before Easter Sunday, he woke up at 3 in the morning, very excited because he knew that his dream was the design of the experiment that was going to prove whether synapses were electrical or chemical. And he scribbled it down on his notepad and fell asleep again. And at 6 o'clock in the next morning, he woke up really excited, but was unable to decipher the notes he'd made on the pad because he'd been half asleep at the time, and was very disappointed. The next night, Easter, he had the same dream. And he woke up. And he was so excited that he didn't write it down this time because he was a bit concerned about that. He ran straight down to his lab at 3 in the morning and he performed the experiment that later won him the Nobel Prize. Good man. So what was this exciting experiment? Well, what he did was he took a frog's heart and he put it in a bath of saline, and he arranged for its vagus nerve to go over an electrode. And the heart would beat in the bath of saline, as hearts do. And when he stimulated the vagus nerve, the heartbeat slowed down. And that's not surprising. People have been doing that for the last 50 or 60 years. It wasn't really very interesting to anybody. But what Lowy did was, he took the effluent from the first tissue bath and fed it into another tissue bath with another frog's heart that this time didn't have an intact vagus. He'd taken the vagus off this frog's heart. And he looked at what happened. And when he stimulated the vagus nerve of the first heart slowed down. But a few seconds later, the second heart also slowed down, despite the fact that it had no electrical innervation with either its own vagus or the first heart. The only connection it had was that it was bathed in the tissue fluid that came out from the first heart. And he concluded from that that the synapse between the vagus nerve and the heart must be chemically mediated, because he was able to wash those chemicals out from the first heart and get them to have an effect on the second heart. And the chemical that he postulated must be responsible, he called vagus stuff, because he was German and it was stuff that came from the vagus, and Germans are logical like that. It later became known to be acetylcholine, and it was the first neurotransmitter to be discovered. So how does this vagus stuff work? We've kind of alluded to this and talked about it in various roundabout ways, and I just want to make sure that everybody is aware of this principle. The parasympathetic nervous system doesn't actually do anything, but it regulates everything else that's doing things. If you take an animal's heart out and disconnect it from its parasympathetic nervous system and put it on a table or in a tissue bath, it will carry on beating for a while. But you won't be able to regulate it and slow its beating down and speed it up. That's what the parasympathetic nervous system does. And it doesn't tell the heart when to beat. The heart works that out for itself. The parasympathetic nervous system regulates that beating and slows it down a bit or speeds it up a bit. And it's very similar to a pilot in an aeroplane. The aeroplane is going in a straight line, very fast if it's one of these aeroplanes, and the pilot is just saying, go that way a bit, miss that mountain, you know, that kind of thing, keeping it under control, but not telling the engines exactly when to come on and how hard to go. He's telling the plane where he wants to go, and the plane is working out how to get there. And so that's the role of the parasympathetic nervous system. It kind of sits back a little bit from the actual tissues, but it controls them. This is a graph from a really interesting paper by Hudson, Mayhew and Pearson. Joe Mayhew is now Professor of Equine Studies here. He took a section of equine small intestine, stuck an intracellular electrode into the smooth muscle cells or into a smooth muscle cell and measured the membrane potential. And you can see that what's happening is there is a regular alteration in the membrane potential of those smooth muscle cells. And that whenever they reach a certain threshold, there is an action potential generated. Now, what the autonomic nervous system is doing is not telling those cells exactly when to get to threshold. It's altering this baseline activity, this regular activity. And in order to study that, Joe and his colleagues added certain chemicals to the bath to alter the way that they worked. The first one they added was a calcium channel blocker, nifedipine, I think. You'll remember that muscular action potentials are mediated by calcium. And when you're trying to put a very fine glass electrode into a muscle cell, its action potential is actually quite frustrating, because it means that the muscle cell jumps off the needle and very often snaps the needle as it goes. And so, although we've actually got a trace here of several pairs of action potentials in a muscle cell, what usually happens is you get a nice membrane potential recording from the cell, it gets an action potential and jumps off, and that's the last you see of that cell. So the first thing to do is to poison the cell effectively with something that stops the action potentials. And when you do that, you get the trace at the top on a slightly different scale. But now we've got those changes in membrane potential without the naughty, annoying action potentials. And you can see that it's really nice and regular. Yeah? There's a regular change in the membrane potentials, and there's two sorts of regularity there. There's the membrane potentials coming along every so often, and there's the general amplitude of the membrane potentials changing, again, in a regular way. They then added tetrodotoxin to the sample. Tetrodotoxin is a neurotoxin, and it prevented the activity of the parasympathetic nervous system in those samples. And you can see now that that regularity is very much disrupted. It's very much changed. And there's much less regularity and much more strange stuff going on, especially round about here in these samples. So that's a really good way of demonstrating that the parasympathetic nervous system is not in charge of making these membrane potentials. These potential changes still happen, but it's in charge of regulating them and deciding how they're going to happen and the patterns. So it's an overall control system. We now need to look at something which some people will tell you is an oxymoron, a sensory autonomic pathway. Because some people will tell you that the autonomic nervous system is exclusively a motor system. I'm deliberately including the sensory pathways in our discussion of the autonomic nervous system because I'm interested in function. And it's the function that I want to get over to you. And it's bizarre and perverse to have a motor system functioning without any sensory input. So we need to talk about how the autonomic nervous system gets its sensation so that it knows what to do with its exquisite motor control. So except for pain, the sensory autonomic pathways are exclusively parasympathetic. And what I mean by that is not that they are parasympathetic, but that they travel with parasympathetic nerves. Okay, so somebody might say that these are autonomic sensory pathways, and somebody else might say that these are sensory pathways which are subconscious but not autonomic and happen to travel with the autonomic nervous system. And whichever of those definitions fits your way of thinking best, that's fine. The vagus, again, carries sensory fibres, mostly stretch receptors, from the alimentary tract, control of gut function, from the bronchi, control of breathing and respiratory function, from the heart and great arteries, control of cardiovascular function. Control there of three of the most important physiological organ systems that there are. The pelvic nerve contains sensory fibres from the descending colon, the bladder, the pelvic viscera, the reproductive tract. So let's have a look at these parasympathetic sensory fibres. And again, you can see that the diagram looks very much like the motor tracts of the parasympathetic nervous system. When we look at pain, it looks different. It's much more segmental and it's more complex. And the reason for that is that visceral pain fibres travel to the central nervous system in both the parasympathetic and the sympathetic systems. So that from the pelvic floor, the reproductive tract, these fibres travel in the parasympathetic system in through the sacral inflow. From the thoracic esophagus cranially, they travel with the parasympathetic system in the vagus nerve. But between those two areas, for the thoracic cavity and for most of the abdominal cavity, they travel with the sympathetic nerves. So it's a more segmental pattern. And that's kind of interesting. If you have a patient under anaesthesia that a surgeon is being unpleasant to in some way, as surgeons tend to, you can get changes in heart rate. And those of you that have experience of surgical practices will probably have seen animals that were anaesthetised, everything's happy, the heart rate's ticking along at 80, A surgeon gets into the abdominal cavity and pulls on something, and suddenly the heart rate's very different. And sometimes it goes up to 120, 150, 160. Sometimes it goes down to 60, 40, 20. The point is that this stimulation is affecting the autonomic nervous system. And interestingly, the areas of the body which are more often associated with a bradycardic response, a slowing of the heart to surgical stimulation, are the areas where these pain fibers travel in the parasympathetic system, the system that's responsible for delivering that bradycardia. So, for example, laryngeal stimulation or stimulation of the upper respiratory tract, cervical stimulation or rectal stimulation, associated with a tachycardia usually, but more often than not associated with a bradycardia, and it can be quite crushing. If you look at the cranial abdomen and the thoracic cavity, stimulation in those areas is more often associated with tachycardia. It's quite uncommon to see bradycardia after surgical stimulation of those areas.